Welcome, everyone, to the Office of NIH History and Stetson Museum Biomedical History Lecture Series. My name is Jeff Resnick. I'm Chief of the History of Medicine Division at the National Library of Medicine. I have the great privilege of introducing our speaker this afternoon, thanks to the very kind invitation of my colleague, Chris Wanchek, whose voice you just heard. Chris is Acting Director of the Office of NIH History and Stetson Museum, with whom my colleagues and I at the National Library of Medicine work very closely to collect and preserve the history of the NIH as part of the broad and deep history of medicine, science, and public health. The Office of NIH History and Stetson Museum Biomedical History Lecture Series provides thoughtful examinations of past success and failures to guide our future. Previous lectures can be seen freely via the NIH Videocast archive, as this one will be in due course, and we encourage you and your colleagues to watch these archive presentations, share them with your colleagues, and of course, look forward to future lectures. So it's now my distinct pleasure to introduce today's speaker, Dr. Robin Wolf-Scheffler. Dr. Scheffler is an associate professor within the program in the Science, Technology, and Society at Massachusetts Institute of Technology. He studies the history of modern biological and biomedical sciences and their intersections with developments in American history. His first book, entitled A Contagious Cause, published in 2019 by the University of Chicago Press, follows the history of cancer virus research in the 20th century, from legislature to laboratory, documenting its origins and impact on the modern biological sciences. He is currently writing a book on the history of the biotechnology industry in Boston, supported by the National Science Foundation and MIT's Leviton Prize in the Humanities. Dr. Scheffler is also in the early stages of preparing a chemical biography of dioxins. The common goal of his projects is to understand the mutual influence of science on society and of society on science. And we're thrilled to have him here with us this afternoon. Dr. Scheffler, over to you, and warm welcome from all of us here at NIH.
Well, wonderful. Thank you so much for that introduction, and thank you for having me virtually as a guest of the NIH, and I'm very excited to speak with you today, and I'm going to share some images because one of the things that I enjoy in my historical work is sort of finding different ways of making the past tangible. And I'll start with the title, but we'll move on fairly rapidly from there. So the topic of my talk today is the relationship between molecular medicine and success and failure through the war on cancer. I'm very happy in particular to be speaking at the NIH at this moment because we're about a week away from the 50th anniversary of the 1971 National Cancer Act, which was signed by President Nixon on December 23rd, 1971. It's colloquially known as the War on Cancer. And the declaration of war on cancer in the 1970s ignites a broad-ranging explosion of research dedicated to both understanding, curing, and preventing cancer. But almost from the outset, it also inspires fierce debate as to whether or not the war on cancer is being won or lost. And indeed, I imagine that some of you who are listening might have even been slightly piqued to see a question mark at the end of my talk title as to whether or not molecular medicine has been a success or failure. The answer, of course, may be so obvious. Molecular biology and molecular medicine have produced so many stunning breakthroughs in the last 50 years against cancer that how could it be anything but a success? But I think actually that thinking about what it means to mark success and failure gets into a number of very interesting questions about what it means to do molecular medicine and the place that molecular medicine has in combating diseases as cancer overall. And I'm going to think about those questions with you in the next 45 minutes or so through looking in particular at the National Cancer Institute's efforts to develop a cancer vaccine in the 1960s and 1970s, an effort which reached its crescendo during the first decade of the war on cancer, and indeed became a project larger than the subsequent Human Genome Project. And remarkably, of course, this effort happened before any human cancer virus was known to exist. Today, of course, we know that there are viruses that are tied to human cancer, but this was not knowledge that existed before the National Cancer Institute poured many billions of dollars into what was seen as a moonshot program to develop a vaccine. And the consequences of this type of spending, both on the possibility of cancer vaccination and understanding the molecular roots of cancer as a whole, really had far-ranging consequences for the development of molecular biology, both within cancer and outside of cancer. Cancer viruses provided a vital path through which molecular biology moved from its first generation, which had largely been focused on studying prokaryotes, bacteria, which are sort of, I won't degrade them by calling them simple organisms, but they are very different organisms than the cells that make up our bodies and are tied to the diseases that we suffer from. And so there's a bridge into eukaryotic cells, the cells that human beings are made up of. And it's really very difficult to imagine that migration without cancer viruses. So there's that migration, which is very exciting, but that migration of molecular biologists also exposes fierce jockeying among activists, administrators, scientists, and doctors over defining what the appropriate aims of the war on cancer should be, and alongside that, what it means to say that molecular medicine has succeeded or failed. And this, of course, is a set of questions that we all have a great deal of interest in answering, not only about cancer, but many other different types of disease. So I'm going to sort of trace that arc, and through this arc, we'll see a number of different moments in which success or failure are discussed both as medical matters, but also as political matters for shaping the future path of research policy. But before I arrive at the war on cancer in the 1970s, I really need to go back a few decades and talk about the one person who, more than anyone, popularizes the idea that research itself is going to be a productive avenue to pursue against cancer, and that is the activist, public relations specialist, art collector known as Mary Lasker. Mary Lasker, who was born Mary Woodward in Wisconsin in 1900, was a New Deal liberal. She moved east to study at Radcliffe and became involved with numerous medical causes after sort of making her first career as in public relations, in particular access to contraception and advocacy for national health insurance during the 1930s. And she and her second husband, Albert Lasker, were both deeply involved in politics. She recalls in one of her oral histories that they spent their honeymoon in 1940 traveling to both Republican and Democratic conventions. And so Mary Lasker is very much sort of within that sort of FDR New Deal liberal tradition of sort of seeing the government as a very effective and powerful agent for improving public welfare. But of course, after the Second World War, a lot of advocacy for national health insurance culminates in President Truman's National Health Insurance Plan in 1948, which is stymied by accusations of state socialism. And this is one of the many cartoons of that sort of appears to oppose it. I find this one especially appropriate given that it's sort of gestures towards the now growing presence of artificial intelligence and various diagnostic strategies for cancer. But in particular, I think that it's interesting too for the types of political lessons that Mary Lasker takes from this setback. Lasker encounters the opposition, which is of course very broad-based because there are doctors in pretty much every congressional district, and doctors as a professional group were not especially enthused about the idea of a state healthcare system. And the lesson that she takes from this is that possibly pursuing federal involvement in healthcare is going to be too difficult. But she pivots based on a shared interest that she and Albert both have in the potential power of research to do something against disease. And this, of course, is inspired by many of the breakthroughs in the early 20th century in microbiology, the development of antibiotics, and of course, in the 1950s, the notable success of the polio vaccine. And Albert and Mary Lasker decide to focus the efforts of the American Cancer Society, which they become major supporters of, on using research to defeat cancer. And research also has the advantage for Lasker of it being a type of activity in which there was no organized opposition. Scientists were generally not people who were well represented in Washington, and Lasker was able to use the connections that she had built and continued to build as a Washington lobbyist to start steering a great deal of money into biological research with the hope that it would produce dividends against cancer and other diseases. And this places the growth of federal support for research on a path to intersect with some of the first articulations of what becomes known as molecular medicine. And this is famously associated with Linus Pauling, who in 1949 publishes a paper, sickle cell anemia, and a molecular disease, claiming for the first time to have identified a particular change in the amino acid sequence of hemoglobin proteins that produces sickle cell disease. And this idea that you could somehow identify the molecular roots of disease carries with it the promising idea that also you might be able to find a molecular medicine to cure those diseases. And by Pauling, accompanied by George Beadle, who also is notable for being one of the people associated with, I think, the one gene, one enzyme hypothesis, begin to appear with the American Cancer Society. They're at a PR event in this photo, advocating for the idea that increased molecular knowledge of life will have beneficial results for understanding cancer. And in the process, they begin to forge these links between government funding for basic science research and improving public welfare through the application of these discoveries. The problem with this vision, which probably sounds very familiar indeed, is sort of integral to the part of the mission of how the National Cancer Institute and the National Institutes of Health begin to understand their work in these decades, runs into some challenges, however. In particular, the mechanism of peer review, which is what the NIH is using to distribute money to all of these university researchers, does not allow for much control over the types of research that are carried out. This is a grant from One Cancer Virus Research term sheet in the 1950s, which literally writes scientific freedom into the terms of the agreement. You don't have to, as a researcher, follow any of the things that you promise you're going to do in your statement. So this immediately makes questions of administration and accountability sort of a ticking time bomb as the National Institutes of Health begins to hand out more and more money through this peer review system, because the odds of it being somehow misadministered become very real. And this is the type of thing that opponents of federal spending on biomedical research, such as Representative Lawrence Fountain, who is a Democrat from North Carolina, seize upon. In the early 1960s, Fountain runs a number of committees that create a great deal of embarrassment for the National Institutes of Health and the National Cancer Institute by pointing out that grants have been mismanaged. So this is sort of a way of getting around the appealing nature of the idea that it might be possible to cure disease at a biological level through a relatively minor investment compared to, say, investing in healthcare. Fountain and others emphasize that the process through which this money is being spent is inefficient and isn't necessarily advancing the good of the taxpayer. And this really is a big problem for the model of basic science support that someone like Mary Lasker is seeking for the National Institutes of Health and the National Cancer Institute. In particular, Fountain and Lambast, the director of the National Institutes of Health, James Shannon, after one particular exchange, that we certainly ought to expect reasonable management procedures to ensure that money is being spent prudently and for the purposes for which it was intended. At the moment, Fountain continued in Congress, we cannot see or measure the results. So this is deeply embarrassing, but it also provides an opportunity for a new way of thinking about what it means for an organization like, say, the National Cancer Institute to oversee basic biological research for the public good. And this is a vision most forcibly articulated by then Assistant Director of the National Cancer Institute, Carl Baker, whose papers were at the Office of History at the NIH, and I think have since been moved to the National Library of Medicine. And I spent a very long summer working through Baker's papers and following Baker really as he goes through this fascinating evolution of his career. He starts off as a trained biochemist working on questions of metabolism, but he has bad asthma. So he's not actually able to spend a lot of time around laboratory animals. And during his time at the NIH, he therefore moves into research and grant administration. And Baker is especially struck in the years before Fountain begins to critique the administration of grants that there might be other ways of administering research that would do a better job of translating biology into useful therapies. And the types of critique that Fountain levels at the National Institutes of Health in the early 1960s really give him his opening to begin pursuing these new structures in a more wide-ranging way. And in particular, he's very drawn to the idea of vaccination against cancer. And although there wasn't a known cancer virus at the time, and I'll talk a bit more about that in a minute, the idea that cancer might be caused by an infectious virus had widespread popular resonance in mid-20th century America. There are outbreaks such as the one in leukemia in Niles, Illinois, that are classified by the Public Health Service as having a possible infectious agent. When a doctor, Paul Levine for the National Cancer Institute, is dispatched to Niles, Illinois to collect blood samples from so-called survivors of the leukemia outbreak, they are so worried about being stigmatized that they are only consent to being photographed with their back to the camera, so they couldn't actually be later identified as possible infectious cancer carriers. So there's a huge amount of belief that based on reports such as these, and also discoveries that there are numerous tumor viruses in animals, that it would be a possibility to find a human cancer virus. And this is where Baker feels that the types of management efforts, ideas that he's been reading about will be especially useful. But this also takes us back to that question of what precisely is a molecule, because the types of viruses that Baker's potentially interested in aren't at the level of, say, a hemoglobin protein. And indeed, for virology, it was only a fairly recent development in the 1960s that viruses had acquired status as molecular entities. One of the famous definitions, of course, of a virus is a virus is a virus is a virus. And you can see that in how the initial discovery by Peyton Rouse in 1911 of what became known, later becomes recognized as the first animal tumor virus, Rouse sarcoma virus, is received. Of course, Rouse is pictured here looking into a microscope, which is slightly ironic in the sense that, of course, you can't see viruses under the optical microscope. And when Rouse was working with extracts from tumors he was getting from chickens from Long Island poultry farmers, he recognized that there was an agent that he thought was transmitting tumors from one chicken to another unfortunate chicken, but he couldn't say precisely in 1910 and 1911 what that agent was. He had no means of visualizing it in the way that you would with bacteria using an optical microscope. He had no means of culturing these viruses in a laboratory outside of chickens. And the only thing that he could say, using what are these are called Birkefeld water filters, pictured at the lower right-hand corner of the screen, is that the types of filtration that would remove larger particles could not remove the hypothetical agent transmitting tumors. So in this rhetoric, viruses were non-filterable agents, things that were too small to be filtered out. But this was not really the type of evidence that was compelling to people either working in germ theory or working in the clinic. Doctors and nurses pointed out that they weren't catching cancer from patients, and Rouse was sort of stymied in this initial identification. And so giving viruses more concrete status as molecular objects is sort of one might think is an important step in mobilizing to find a vaccine against them. And of course, vaccines as a whole could be considered a form of molecular medicine in that regard. And this is the type of work that often you could think of as the electron microscope as providing a valuable resource for doing it. And of course, the argument here is that the electron microscope is able to help us visualize viruses in the same way that we might look for microbes using an optical microscope. And indeed, by the 1950s, advocates of the electron microscope as a means of tumor virus hunting, such as the Polish-American researcher Leon Domachowski at MD Anderson in Houston, begin to claim that they have identified virus particles in sections of breast cancer tissue from both mice and humans. The Boston Globe writes that Domachowski has managed to put viruses at, quote, the scene of the crime. But this type of evidence, although it gives viruses that particle-like nature that we might associate with them, is not compelling necessarily to even other virologists at the time. I've tried to helpfully circle what Domachowski claims is a virus particle in his slide image, his electron micrograph on the right. But another virologist looking at images like these at a conference in the 1950s pointedly reminds Domachowski that images are unlabeled underneath the electron microscope. So there's still a great deal of interpretation and dissent among virologists and cancer researchers as to whether or not these proofs that there are cancer viruses are actually compelling, and certainly whether or not there's anything like that that's been identified in the case of human cancer. And this is where Baker's reading and management theory becomes actually very important. The type of approach that Baker proposes, which is illustrated here in a publicity still from a few years later, focuses on a new way of thinking about research drawn from the RAND Corporation of Santa Monica and developed into what's known as systems analysis at the Defense Department in the early 1960s. And these management methods were designed to address the urgency of Cold War research and development in competition with the Soviet Union, and also to manage sort of complex projects in which there were multiple elements that might or might not be fully present at the beginning of a project, which seemed like a very useful way of thinking about developing a cancer vaccine, because it said that the question of whether or not there was a human cancer virus could be one of many things that the National Cancer Institute could begin to work on. And coupled with that is this very potent vision you see for discussing cancer viruses in paperwork and in administration, which emphasizes anticipated results or the inevitability of certain developments. And you can see this in the types of annual reports that the Special Virus Leukemia Program, which Baker spearheads, starts to work out. You can see it here in these dotted lines from the different stages on the left-hand side of what you go through for developing a vaccine, from acquisition of materials through detection, isolation, replication, identification, characterization, industrial replication, and control. And you can see that these, of course, have been successfully carried out for polio and measles in humans on the right. Indeed, the claim is that this has already happened for certain animal models for leukemia or lymphoma viruses. And really, the dotted lines you see for humans highlighted in red are evidence of things that are about to come. This is also part of the logo of the program that's released a year later, which emphasizes here, if you see those sort of hexagons at the top representing viruses and the sort of the hatched arrow to humans, that's the last causal arrow to be filled in the types of things that cause cancer in animal and human models. And this type of framing of what cancer viruses are is very far from the types of laboratory realism that are associated with the typical path of vaccine development. But they are nonetheless extremely powerful in mobilizing resources at the National Cancer Institute for research into cancer viruses. The rhetoric here very much, as Kenneth Endicott, then the director of the National Cancer Institute, explains to Congress, not a question of if a virus would be found, but when it would be found and how quickly a vaccine could be developed using that knowledge. And Baker is also a fan of rising at conferences and reminding people how many people, attendees, how many children have died of leukemia during the days of the conference. And so there's a strong element of urgency that this type of management framework claims to be able to address. And indeed, the argument is that until you invest in this infrastructure, you're not going to be able to find human cancer viruses, because a lot of the work that needs to be done isn't necessarily high-tech scientific work, but it's really sort of large-scale collection and screening. But that brings immense resources. Very soon, the National Cancer Institute is spending 25 times as much money on cancer virology as the American Cancer Society. They build a whole set of laboratories in Building 41, the National Cancer Institute, which are biohazard containment facilities designed to handle these potentially oncogenic human viruses when they are discovered. And so this is really a testament to the types of powerful ways of managing molecules that you can see. And of course, this was something that I was extremely puzzled by when I started reading about it in Baker's archives many years ago. But what I was especially perplexed by is what happens next, which is that after having this program in operation for about 5 years and having lots and lots of resources and searching diligently for human cancer viruses using methods from immunology, using methods from electron microscopy, the program has yet to actually find any evidence that there's a human cancer virus that you could target for the development of a vaccine. And this is where a really fascinating turn happens once again. In particular, Robert J. Huebner, who's a famed vaccine developer at the National Institute of Allergy and Infectious Disease, who moves over to the National Cancer Institute to help with the what becomes known as the Special Virus Cancer Program, as it expands from leukemia to all cancers, begins to think that the challenge is not in the failure to find a cancer virus, but that they've been thinking about what viruses are in relationship to the causation of human cancer all wrong. And in 1969, he publishes a paper with George Todaro, trying to explain why the viral hypothesis for human cancer remains true, even though all of the methods that virus hunters typically use have yet to actually reveal the human cancer virus. And they call this the oncogene theory, or the viral oncogene theory. And this is really a creative attempt to put together insights from the study of phage, which are viruses that prey on bacteria such as E. coli, with what their theories of genetics are. And I've sort of put this together, this illustration, to give you a sense of what happens. And the idea is that a standard virus, which is sort of the rectangle up top, has genes, virus genes, which are responsible for replicating a virus, that are put into a healthy cell. And after that round of infection, that healthy cell will lysis, or burst open, and release all these fragments of new virus that contain virus genes. However, a certain special kind of virus, what Huebner focuses on, are called types, C-type RNA tumor viruses, which go by the slightly snappier name retroviruses later on, are a certain type of virus that contains something called an oncogene, which is sort of shaded in black here. And that oncogene is inserted into a healthy cell along with the virus gene. And in many cases, the cell itself does not get burst open by viral infection. Instead, the cell continues to reproduce with the oncogene present, and only in certain instances, such as with that asterisk mark, it could be radiation or some other type of chemical mutagen, or just random chance, do you see a cell become cancerous. And so this is a way of preserving the idea that cancer is caused by a virus, while also explaining why it's so difficult to actually see virus at the scene of infection. And this is something that in Huebner's mind also explains the phenomena that many animal cancer virus researchers had seen, which is called vertical transmission. So this is a theory that does sort of seem to neatly encapsulate what Huebner called a contradiction that had to be resolved. And of course, this theory has a few challenges. Principally, in 1969, it's a theory that suggests that RNA viruses are able to edit the DNA of cells, and there is no chemical mechanism known at that point by which that's possible. Indeed, the idea of Huebner's oncogene theory seems to run against what's called the central dogma of molecular biology, here sort of summarized by Francis Crick, which is that if you think about molecules as having information flow, the flow is always from DNA to RNA to protein, and never upstream from protein to RNA to DNA. So the viral oncogene theory hypothesis that Huebner puts together seems to not only be a form of spectral fleeting to explain the types of setbacks that the special virus cancer programs encountered, but also biochemically implausible. However, as some of you may be thinking, in 1970, quite dramatically, two scientists, Howard Temmett at the University of Wisconsin-Madison and David Baltimore of MIT, both announced roughly concurrently that they've both identified an enzyme, reverse transcriptase, that can allow the types of RNA editing of DNA in cells to occur. And this discovery, which seems to occurs right as lobbying for the war on cancer begins to kick off, is seen as a unique validation of this idea that it is actually possible to plan research, that it will be possible to predict the path of research and devote resources to developing it, rather than to simply await results in the laboratory. In the words of one advocate for the NCI's super plan to develop a cancer vaccine, it will do more than give out money and wait for results. It will plan research and make results. And this is something that is sort of a core idea of the war on cancer, as it's articulated, is that the war was not only going to increase spending on cancer research, but also bring the types of planning methods that Baker had developed for cancer vaccination to bear on a wide variety of problems of cancer research. And indeed, when the war on cancer is declared, the amount of money flowing into the National Cancer Institute's vaccination program explodes, and the investment in retroviruses increases the number of people working on retroviruses almost 100-fold between 1970 and 1974. creating what an editorial in Nature complains was almost an instantaneously overcrowded field. And two of the researchers who are drawn into that overcrowded field are University of California, San Francisco, J. Michael Bishop, and of course, someone who may be very familiar to those of you from the NIH in the audience, Harold Varmus. And these were both individuals who had not necessarily made cancer research initially their primary focus. J. Michael Bishop had worked on polio, which is another RNA virus, and of course, Harold Varmus had worked, had been trained as a doctor and had done his research fellowship at the NIH and had become interested in the lac operon in E. coli. But the amount of money that's floating around for research onto the viral oncogene draws them into what's known as the West Coast Tumor Virus Cooperative, which is a set of groups, some University of Washington, Seattle, University of California, Berkeley, University of Southern California in Los Angeles, that are all trying to understand and identify a possible viral oncogene associated with Rous sarcoma virus. And this is a community that really allows a very dramatic discovery. You can sort of see some of them gathered together at a Rous lunch. The core question that Huebner and other members of the National Cancer Institute are interested in is designing a probe that will allow researchers to demonstrate that there are actually these viral oncogenes in cells that are turning cancerous. And it turns out that there's this very sensitive technique known as Rna Dna Hybridization that Bishop and Varmus begin to master, and they begin to sort of go hunting for the oncogene or they think might be the oncogene in RSV, which I would like to stress was just a fantastically expensive process. For example, by 1975, the cost of radioisotopes alone is more than the salary of a skilled laboratory technician. And this type of work sort of creates a large four-member group, all laboring at the University of California, San Francisco, including 4 sort of lead professors, 10 support staff, 15 postdoctoral fellows, graduate students, and other professors who come in. And this allows the manufacture of this very sensitive, but also very perishable radioactive probe to look for a viral oncogene. And I sort of stress this because without all these resources, it's very difficult to imagine what happens next happening. And the general idea is here, which is that if you can use reverse transcriptase and hybridization with an RSV mutant, it's possible to merge all the parts of Rous sarcoma virus that don't contain an oncogene and leave this one oncogene containing fragment unhybridized, which then you can hybridize with DNA from normal cells to hunt for chromosomal oncogenes. And this is a process that once you start investing it, you sort of need to keep carrying out. And the fascinating thing is that the identification of the oncogene using this probe is successful in 1975. But what's truly remarkable is what happens when they start to run the calibration experiments. These are supposed to be routine experiments intended to reveal that the oncogene probe does not find cancer-causing genes in cells that are not cancerous, but instead, as Bishop and Varmus and their postdoc their co-researcher Dominique Stellian report in January of 1976 in Nature, it actually turns out that the hybridization of this probe with DNA from so-called normal cells is substantial. And so what had been thought of as a viral oncogene is actually already present in the genome of normal cells. And so what this leads to is a dramatic reinterpretation, illustrated by Jamie Simon here from a few years later, that it's not actually the retrovirus that gives an oncogene to a healthy cell. It's actually an innocent retrovirus that during a recombination event at some point in the past becomes oncogenic by picking up a cellular oncogene. And Bishop and Varmus spend quite a bit of time the next few years sort of using different degrees of hybridization to show that there must be a type of evolutionary divergence, and sort of going back and there's sort of at some point in evolutionary time, there had been this moment, which explains why there's better and worse degrees of overlap in the probe with different types of, if you go back to this chart of results, for example, things that are closely related to chickens, such as quails and turkeys or ducks, have a good degree of hybridization overlap. But when you get to the emu, which they call a very primitive bird, you see much less. And then when you get into mammals, you see far less. So this is a dramatic reorientation of the aims of the NCI's cancer vaccination program. And this discovery, though, that there are cellular oncogenes would have been very difficult to accomplish without all the resources invested in this probe. It rapidly becomes, as you can see from all the other caricatures of different researchers here, a rapidly growing field. And ironically, this is the risocoronavirus is still the easiest way to study these types of oncogenes before the development of polymerase chain reactions or recombinant DNA in the mid 70s. And indeed, other workers trying to do similar experiments using other virus systems, such as Ed Skolnick at the National Cancer Institute, working with murine sarcoma virus, find the work so frustrating that a research group, according to Skolnick at least, threatened to quit in 1975. So it's a combination of a lot of resources and a bit of good luck, but all within the context of this search for an external agent for cancer that suddenly, through the oncogene probe, becomes redefined as a cellular oncogene, an enemy from within, if you will. So this is one of these moments where the infrastructure does create a new discovery. And the question, of course, is, does that count as a point of success or failure for the original aims of the war on cancer? And this is a question that, returning to that moment in the early 70s, is a deeply political one. The declaration of war on cancer really brings the national and the excitement that Huebner and others inspire in sort of the promise of molecular biology, brings the National Cancer Institute and the war on cancer into direct contact with a broader community of molecular biologists, of whom James Watson is sort of the most visible sort of representative in the first few years of the war on cancer. But it's also a tradition based on sort of what's known as the Phage School, which is a much more freewheeling school. These are a group of scientists who, in many cases, got into biology to escape some of the heavy-handed state planning that they saw associated with the rise of nuclear energy and the atomic bomb project, a physics of life as they thought of it, rather than physics of death. And they see themselves as creative, curiosity-driven experimenters. And the types of research planning that the National Cancer Institute is proposing seem to not only be ineffective in their eyes, but actually really a threat to the type of scientific community and scientific continuity that they would like to have. And the challenge, as someone like Watson very quickly figures out, is that due to an economic slowdown, the expense of the Vietnam War, there's not as much money starting in the early 1970s in these other kinds of research as there is in cancer research, which is much more organized. Here I'm just using the two budgets of the National Institute of General Medical Sciences and the National Cancer Institute. And you can see that although they were roughly comparable through about 1970, cancer research really becomes a much more substantial amount of money. Then there's a concern voiced by Watson and others that this type of emphasis on the clinical applications of molecular biology is going to cause safe but sort of intellectually uninspiring work. Arthur Kornberg from Stanford charges that cancer research will seduce young scientists away from more important fundamental work. And so one of the things that's happening alongside Bishop and Barmas's work is a fierce debate spearheaded by Watson, but also one of his colleagues, Norton Zinder, to redefine what the purpose of cancer research should be. And they take the cancer virus program at the National Cancer Institute as their primary target, as a case for why the government shouldn't be allowed to manage molecular biology. And here, of course, the claim is that you need to have an understanding of the fundamental biology of disease to make any meaningful headway, and that fundamental biology is best obtained through peer-reviewed research, as opposed to the types of research planning that the National Cancer Institute is seeking to implement. This comes to a head in a controversy over a report which is sort of leaked to the press by Norton Zinder, which is very critical of the National Cancer Institute and the what's then called the Virus Cancer Program, essentially asserting that without being attentive to the needs of scientists, it's going to be impossible to develop a cancer vaccine. Watson has a pointed editorial making all these parallels to the Vietnam War, actually, saying that the government's wish for instant victory shouldn't be allowed to divert scientific work. Although it's also very interesting that he also manages to say that the government is both fighting a war in Vietnam, misguidedly, but also that it's acting as a commissar. So there's basically every single type of derision that Watson can heap on planning is present in this editorial. And so by the mid-1970s, these molecular biologists have really dramatically changed the conversation about the war on cancer, sort of defining it as a failure precisely for its inability to support the types of creative molecular biology that they think are appropriate. However, this looks very different if you're not in the molecular biology community. And indeed, by the late 1970s, there's a great deal of disillusionment with the war on cancer as a whole. Part of this is motivated by the fact that rates of cancer incidence continue to climb through the 1970s, and also, of course, that rates of success in cancer treatment remain fairly stubbornly stable in most cases. So by the late 1970s, you begin to see reports of disillusionment. Most infamously, the outgoing director of the Food and Drug Administration calls the war on cancer a medical Vietnam. And this type of dissatisfaction opens the National Cancer Institute and federal spending on cancer research to a whole other set of critiques from different groups. Civil rights groups begin to sort of ask for more attention to the relationship between cancer and racism. The women's health movement begins to ask for more attention to breast cancer and a different emphasis on assessing different types of screening and treatment. But most critically, for cancer research at the National Cancer Institute is the rise of the environmental movement, anchored, of course, in works such as Rachel Carson's Silent Spring, which, despite largely being remembered as a book about the effect of pesticides on nature, contains quite a bit about carcinogens and the causes of cancer. Here, of course, this is casting cancer as a disease of civilization. Carson warns that we are living in a sea of carcinogens, and this is a very different view of what it means to be at risk of cancer than oncogenes. This is from an earlier book, but you can see all of these different things that also could be thought of as molecules, converging on the human body to sort of cause disease. And this critique is forcibly articulated by Samuel Epstein in the late 1970s, and of course summarized by the great Herb Bloch as an editorial cartoon, which is to say that actually the act of looking in the laboratory for trying to cure cancer is a distraction from the multitude of environmental causes that are to be found in society, and really the war on cancer should be waged against those causes as opposed to focused on work in the laboratory. And in the hands of Epstein's critique, all of the critiques that the molecular biology community had leveled at the National Cancer Institute's cancer virus program are actually critiques that aren't just about virus research or planning, but they're actually indictments of the entire process of using basic science to try to do something about cancer. Epstein charges during congressional testimony that most research was irrelevant to cancer treatment or cancer prevention. And you begin to see, of course, a renewed emphasis on these sort of environmental causes in California's Proposition 65 and within popular culture. And you also begin to see it in the budget of the National Cancer Institute, which does experience this, begins to experience this sharp drop right before 1980. And this is sort of really a message that sort of the war on cancer shouldn't necessarily be fought through this type of investment in research or even focusing on therapy. There's a whole world of prevention and occupational health that needs to be addressed. And actually, the eroding fortunes in budgetary terms of the National Cancer Institute were only amplified by the arrival of the Reagan administration in 1980. And although, of course, President Reagan was not really known for his zealous advocacy for environmental regulation, he also was perfectly comfortable with retrenchment in budgets for civilian research. So in the early 1980s, the budget for the National Cancer Institute appears to be under threat. And with that, of course, molecular biologists and biomedical researchers at universities and hospitals throughout the country begin to be concerned that once again, the overall health of the scientific community is going to be under threat because of the decline in funding for cancer research, which is such a dominant part of the molecular biology community. The irony, of course, is that someone like James Watson, who's the director of Cold Spring Harbor Laboratory in Long Island, actually, despite his criticism of the war on cancer, really draws upon the National Cancer Institute and cancer virus research, in particular, very heavily to support the laboratory's budget. So it's in this sense that I want to come back to the politics of what it means to discover oncogenes and what happens afterwards. One of the interesting features of the conversion of retrovirus research into oncogenes is not only that, in the words of one scientist, that work that had appeared to be stamp collecting, sort of collecting different types of retroviruses, actually became this very potent tool for exploring the genetics of cellular growth and development, but also it happens so rapidly. If you look at these timelines, it's just a very, very rapid unfolding of events between about 1979 through 1984 in particular, which happens to layer precisely onto this moment of budgetary and political crisis that the National Cancer Institute is facing. And in particular, Robert Weinberg, who's a biologist at MIT, develops a new assay for oncogenes that doesn't require retroviruses in 1982, and this produces the identification of the first potential human oncogene, RAS. And the question then is what to do with this new promise of research focused on cellular oncogenes in light of the fact that most molecular biologists had spent the previous decade being deeply critical of the National Cancer Institute. So how are they going to find a new way of existing together? And what historical account will be given that sort of makes that possible? And this is something that Watson and the new director of the National Cancer Institute, Vincent DeVita, managed to sort of do in this interesting sort of set of choreography in 1982, right in the aftermath of Weinberg's discovery. That summer, Watson invites DeVita to speak at the dedication of the Reginald Harris Building at Cold Spring Harbor Laboratory, which would support work on mammalian genetics and potentially the genetics of cancer. And Watson times this meeting to occur right after the annual meeting at Cold Spring Harbor on RNA tumor viruses, which at that point had roughly 400 scientists. And so the timing of this is supposed to really suggest a strong amount of continuity between this type of virus work and the new promise of work in molecular genetics. And at this dedication, DeVita gives a speech, which is later republished in Cancer Research in a modified form, extolling the war on cancer for the serendipitous contributions it made to the growth of the oncogene theory, sort of recasting the research planning efforts not as deliberate, but as a matter of good luck, and therefore saying that there's a brand new sort of therapeutic frontier opened by oncogene research, and that the real danger is of failing to pursue and apply one of the most exciting areas of research that brings molecular biology to the crowded bedside of the cancer patient. A good dose of optimism seems about the right sort of tone to strike. And of course, excuse me, this is very much addressing the concerns that the budget for the National Cancer Institute is going to be cut. And in this speech, he quotes Watson praising the power of the public purse in the war on cancer, which is a dramatic change in opinion. And this sort of mutual moment of recognition sort of moves to refocus attention on oncogenes. It's a famous meeting that's set up for oncogenes outside at Hood Carroll's, Maryland, supported by the Frederick by the NCI, and the focus on the possible therapeutic payoff of oncogenes becomes front and center and sort of stabilizes the sort of that moment of doubt in the sort of the molecular medical approach to cancer. And of course, you see this, the idea of oncogenes, hypothetical or not, becomes sort of a new basis for underlining and underscoring the promise that molecular biology has for therapies against cancer. But I would highlight that this sort of wholesale realignment, this praise of the therapeutic possibilities of oncogenes in the 1980s is really still very much ahead of their whatever therapeutic benefits they may have had. So calling them a success or failure in that moment is still something that's very much tied up with the politics of positioning molecular biology and molecular medicine rather than its actual accomplishment. So in that sense, it's similar to the future orientation of someone like Carl Baker. And you can see this too. One breast cancer activist, Rose Kushner, bitterly wrote that she visited the National Cancer Institute in the late 1970s, hoping that vaccination against breast cancer was imminent, that it would be declared any day dawn. Instead, I had learned that cancer may be nature's own solution to the population explosion and inborn suicidal oncogene. And so this type of promise and this type of response brings me back to the opening question of my talk, which is, to what degree do we see molecular medicine, the war on cancer, as a success or failure? And as a historian, I just have to say this really depends on who you empower to ask that question and who has the authority to answer it, and the concerns that they have will they do. It's certainly the case that the war on cancer has had an outsized impact on the growth of molecular biology. The other question, however, to ask is whether or not molecular biology has had an outsized impact on the cancer problem writ large. And just to capture some of that, I want to go back to the late 1970s for one sort of concluding story that really captures the essence of how this question of molecular medicine success or failure both illuminates and obscures some of the broader questions we might think about when it comes to what type of problem cancer is. And this is a moment in February of 1978 when the new director of the National Cancer Institute, Arthur Upton, gathers a council of noted scientists to sort of help him think about the trajectory of the war on cancer. And it includes David Baltimore, who got a Nobel Prize for the discovery of reverse transcriptase, and Baruch Blumberg, who also was a Nobel Prize winner for his work to identify the hepatitis B virus. And right before this meeting convened, there was some of the first reports that hepatitis B infection was linked to liver cancer were reported, and it was seeming increasingly possible that it would be actually feasible to develop a vaccine against hepatitis B. And the exchange that Blumberg and Baltimore have that day really exemplifies exactly sort of some of the both the successes and shortcomings of molecular medicine. Baltimore says that it really doesn't make any sense to try to think about the hepatitis B liver cancer link without understanding the molecular mechanisms at work in the process of infection and oncogenesis, whereas Blumberg, who's trained in immunology, responds that it was fairly customary to develop vaccines without understanding the molecular mechanism of infection. But what's really remarkable to me after that is Baltimore's retort is that even if you were to cure all the liver cancers in East Africa, we would not have responded to the mandate to deal with the problems we have here in the United States, instead of studying ways of preventing cancer anywhere in the world. And that comes back to the fact, which was the case, that the focus of the National Cancer Institute was on the cancer problem as it was understood in America, and the hepatitis B liver cancer link was something far more prevalent in East Asia and Sub-Saharan Africa. But this retort also really sort of shows some of the larger assumptions that we make when we talk about succeeding or failing. In this case, of course, from whose perspective and whose national perspective we stand in. I think at this current moment when we're sort of thinking about what it means to say succeed or fail and protecting people against COVID-19, that's a memory to have in mind. And finally, of course, like a common refrain is that we should fight cancer with knowledge. It's been something here for the entire 20th century. And I guess my big thought is the history of how we've talked about success or failure is part of that knowledge we can bring to bear on the next 50 years of cancer research and our attempts to control and prevent it. Thank you very much. I'll be looking forward to taking a few questions.
Dr. Scheffler, thank you very much for a fascinating talk. We do have a couple of questions, and we'll jump right in. The first is, given that we know now that cancer is not a single disease, do you think that the war on cancer was scientifically naive, or rather deliberately simplified with a catchy focus in order to secure the funding?
Yeah, this is a critique that I see frequently, and there's, I think, equal parts politics and intellectual ambition in thinking of cancer as a single disease. On the one hand, it's much more evocative to talk about a disease that touches, say, the lives of 1 out of every 2 Americans, rather than dividing cancer up into the hundreds of different categories that you could classify it as. But equally, too, at least for large parts of the 20th century, you have had that ambition that there would be one basic biological key that could unlock something fundamental to the disease. And you see definitions of cancer such as cellular dysregulation or the idea that single point mutations in a set number of genes can give rise to cancer, as a way of trying to take what appears to be a very diverse disease category and actually say, no, it can be identified through a small number of mechanisms and therefore treated that way. And of course, there's the tension exactly between those two different ways of framing cancer, something that's so large that it needs mobilization versus something that's so fractured, it's hard to imagine how to mobilize any one set of approaches against it.
Thank you. Another question that's come through is, do you see a parallel history evolving with the current cancer moonshot? Perhaps related, are we still in the war on cancer, or did the troops, or the funding that is, pull out at some point?
Yeah, that's a wonderful question. I certainly think with President Biden's, first when he was vice president, his work on the new cancer moonshot, which I very deliberately I think has the word moonshot rather than war, because sort of the war metaphor has become a source of disillusionment. And of course now with ARPA-H, you're beginning to see the same questions emerge in terms of not only what types of research do we need to carry out, but what forms of research are appropriate and how should it be organized. So in that sense, I don't think the war on cancer has ever really ended. And of course, how could we declare the war on cancer over when cancer is still such a major presence for Americans and throughout the globe? But I do think that the degree to which cancer becomes seen as the problem waxes and wanes. And this gets into these questions that military historians also ask, is like, what does it mean to end a war? And that's, of course, a much more complicated process than we often think.
Understood. Can we take a step back? Your thoughts just a moment ago about the shift from war on subject to moonshot. Can you say a little more about how you've observed this shift in a broader ecosystem? I mean, there's been multiple wars on drugs, on various medical conditions. Do you see an overall shift away from the war on metaphor to other things, like you suggested with regard to cancer? Just curious.
I think I do. And I sort of came into the history of cancer having read people like cultural commentators like Susan Sontag, who were very critical of the war metaphor. And indeed, I don't think it's necessarily the best metaphor if we're talking about any type of disease. It sort of implies, of course, that people who fall sick from cancer are somehow losing because they're not fighting enough, which is not an appropriate implication to make by any stretch of the imagination. But nonetheless, you can see the power of the war metaphor historically. You have the war on poverty, you have the war on cancer, you have the war on drugs, you have the war on terror. And those are none of those wars especially end in victory the way that, say, World War II does. And they sort of all encounter these moments of frustration. And I do think, yeah, especially in the last several, maybe the last 5 or 10 years, you start to see a renewed sense of frontier metaphors and space metaphors. So you've had the moonshot, warp speed even for the Trump administration's push into vaccine development. These are all very different sets of imagery. And I think that it's precisely because they have that idea of like outward expansion rather than victory. They sort of maybe are they're a bit more flexible and possibly less politically fraught because no one can ever really accuse you of losing a moonshot. They can just sort of, say that you should reach higher.
Thank you. Very good thoughts. So another question's come in. Even before Carl Baker's ideas, this person writes, we had Ludwig Gross and others who had identified potential cancer viruses. Did this not contribute greatly to initiating the war on cancer?
Yes, and Ludwig Gross is a fascinating character. He's a Polish Jewish virologist who has to at one point flee from the Pasteur Institute in front of the invading Nazi armies, but keeping some of the mice that he wanted to work with on his person, and he kept sort of worked with them in his car when he was at the Veterans Administration in the Bronx after the Second World War. And so, yeah, there are. That's exactly one of the disconnects I find so interesting, is that there are people who are working on animal models of viral carcinogenesis, and they are identifying a number of potential viruses, but that type of scientific evidence doesn't really ever get to the level of inspiring a type of super plan or a sort of a full push for vaccination, because the relevance to humans isn't something that's trusted. And that's where someone like Baker is interesting, because he sort of figures out a way of talking about research and about vaccination that makes the leap far before many sort of clinicians would have been willing to sort of make the leap.
Thank you. Thank you. We're going to leave it there. It's 2 o'clock with respect to the scheduling. Dr. Scheffler, thank you very much for a fascinating talk, and both from all of its content, but especially visually in terms of the source material that you've shown. And I want to just mention that by way of offering, again, thanks from all of us at NIH and from the Office of NIH History and STEP Museum, especially the sponsors of this talk. And you're at MIT, as we all know, so folks can reach out to you with any questions, and I'm sure perhaps they will. And we wish you all the best in your continued research and your future projects. Thank you.
All right. Thank you very much, and thank you for listening. And I also want to just extend a deep thanks to both the National Library of Medicine and the NIH Office of History for making a lot of the research beneath both these images and the content of the talk possible. So thank you very much, and good afternoon.
Thank you. Likewise.